In this presentation, let us discuss some typical polysaccharides. Polysaccharides are polymers of monosaccharides, which could be homo or heteropolysaccharides. They can serve structural and energy storage functions. Among the most common homopolysaccharides in animals and plants are starch, glycogen, and cellulose. Starch is made of amylose and amylopectin molecules. The former are usually unbranched, short polymers linked by alpha-1,4 glycosidic bonds, and the latter are larger branched chains linked by alpha-1,4 and alpha-1,6 linkages. Glycogen is similar to amylopectin but have shorter chains with more extensive branching. Cellulose is a polymer of glucose units linked by beta-1,4 linkages and beta-1,6 bonds. Both starch and glycogen have storage functions, and cellulose serves important structural roles in higher plants. Examples of heteropolysaccharides include glycosaminoglycans and murin, etc. Let us now discuss the structure and functions of starch, a storage polysaccharide in plants. Starch is a naturally occurring plant storage carbohydrate, mainly found in the seeds, fruits, tubers, roots, and stem pit of plants, notably in corn, potatoes, wheat, and rice. Two polysaccharides occur together in starch, amylose and amylopectin. Amylose are usually unbranched chains of D-glucose residues linked with alpha-1,4 glycosidic bonds. Amylopectin is a branched polymer containing both alpha-1,4 and alpha-1,6 glycosidic linkages. The alpha-1,6 branch points may occur every 20 to 25 glucose residues to prevent helix formation. Starch digestion usually begins in the mouth. Alpha amylase in the saliva initiates hydrolysis of the glycosidic linkages. Amylose structure. Amylose, as we have seen before, is unbranched polymer of glucose units linked by alpha-1,4 glycosidic linkages. Functions of amylose. Amylose plays important roles in plant energy storage. It is less readily digested than amylopectin. Amylose is more linear, that is, less branched than amylopectin. Therefore, it is more compact and occupies less space than amylopectin. So, it is the preferred form of storage molecule in plants. Amylopectin. Amylopectin constitutes about 80% of starch. It is a polymer of glucose residues linked by alpha-1,4 glycosidic bonds. It is more highly branched than amylose. Almost every 25 glucose residues in the main chain of amylopectin, there is a branch point of glucose units linked by alpha-1,6 glycosidic bonds. Structure. Let us look at the structure of amylopectin once again. Glucose units are linked by alpha-1,4 bonds with alpha-1,6 bonds at the branch points. Functions of starch. Let us highlight the major functions of starch. It is the major form of storage carbohydrate in plants. Starch is stored in specialized organelles called amyloplasts. When energy is needed for cellular work, plant cells hydrolyze the starch, releasing glucose, which is a metabolic fuel. Humans have enzymes that can digest starch and release glucose as energy source. Starch is the principal dietary carbohydrate for humans. It is the major source of metabolic fuel, that is, glucose. Glycogen. Let us look at the next typical polysaccharide, which is a homopolysaccharide, that is glycogen. Glycogen structure. The structure of glycogen resembles that of amylopectin, but with shorter chains and greater branching. Functions of glycogen. Glycogen is the major form of storage carbohydrate in animal liver and skeletal muscles. It is the major secondary short-term energy storage molecule in animals. Primary storage molecule is fats. Glycogen forms an energy reserve that can be quickly mobilized to meet a sudden need for glucose, for example, during sudden running action. Only liver glycogen can be made accessible to other organs. Liver glycogen supplies energy during starvation. Muscle glycogen provides metabolic fuel, that is glucose, to muscles only during exercise. Let us now compare the structures of amylose, amylopectin, and glycogen as can be seen in the accompanying figure. Let us now look at the structures and functions of cellulose molecules. Both starch and cellulose are homopolymers of glucose, but the difference between the two types of polymers is in the type of linkages between the glucose residues. In the former, the bonds are of alpha-1,4 type, while in the latter, it is beta-1,4 glycosidic bonds. We can look at the differences between the two molecules in the accompanying table. Functions of cellulose. Cellulose has major structural role in plant cells. Cellulosic cell walls provide high tensile strength to plant cells. It provides rigidity to plant cell walls. It has a high density of hydrogen bonds which prevent water from entering the molecule. So cellulose is usually resistant to hydrolysis by cellulolytic enzymes. Wood is largely cellulose. Cotton and paper are almost pure cellulose. Cellulose in food provides roughage, increasing intestinal mobility, and is largely eliminated unchanged. Conclusions. Cellulose and amylose can be compared in terms of their structure and functions. Both cellulose and amylose consist of polysaccharide chains containing glucose residues bonded together with glycosidic bonds. Both molecules are insoluble in water. However, cellulose is composed of beta-glucose and forms long straight chains in which every other glucose monomer is flipped over and are very strong, whereas amylose is composed of alpha-glucose residues, forms a coil chain, and they tend to be found in granules. Because of their structure, cellulose and amylose have very different functions. Amylose is used as an energy storage in starch, whereas cellulose plays a structural role. Starch, glycogen, and cellulose are all polymers of glucose. They differ in the type of glucose present and the bonds which link their glucose monomers together. Starch and glycogen are made from alpha-glucose residues. This is an isomer of glucose in which the hydroxyl group attached to carbon atom number 1 is below the plane of the ring. In this presentation, we have discussed the structures and functions of three major homopolysaccharides, that is starch, glycogen, and cellulose. I hope that it was a gratifying learning experience for you. Good luck for your further academic odyssey.